So today we're going to start with autonomics. So when you think about autonomics, I want you to consider the fact that these are the neurons that are controlling involuntary motor activity. So we're not talking about sensory, we're talking about involuntary motor. So what cell types are responsible for involuntary motor activity? Smooth and cardiac muscle, right? So we're talking about the control of smooth and cardiac muscle in the body. And there are very limited numbers of tissues that actually have smooth and or especially cardiac cells that make them up. So the autonomic nervous system is basically your involuntary part that controls these organs. And what you actually have is this competing activity between the two aspects of the autonomic nervous system, parasympathetic and sympathetic. So what I always do is start with the parasympathetics first because we've just done cranial nerves. And it's important to recognize that 4 of the 12 cranial nerves have a parasympathetic component. That's absolutely critical. It's also important to recognize that cranial nerves do not have any sympathetic function. It's only parasympathetic fibers that travel in 4 of the 12 cranial nerves. So when you look at the autonomic nervous system, what we're going to see is, in fact, there are some similarities, and there are obviously some differences between parasympathetics and sympathetics. And some of these differences are really straightforward, okay? What I really want you to get a grasp of is the basic function, what the parasympathetics do in the cranial nerves, and we'll talk about a few sympathetics, aspects of sympathetics that are very important. So again, I come back to this. There's so many different textbooks out there with so many different artists' renditions of the autonomic nervous system. But this to me is like the best. You know, it's clear, it's concise, it has everything on there that you want, no more, and certainly no less. So I hope you use these. I know you've probably been given other ones, and you will be given other ones, but I hope you end up using these because I really think that they're the best ones. So when we think about the parasympathetic and sympathetic nervous system, come on, guys. That's all right. When we think about these two parts of the nervous system, we can consider the fact that they're made up of 2 neurons. And these neurons get referred to as being pre- and post-ganglionic. So if a ganglion is a collection of neurons situated outside of the central nervous system, then somewhere in the organ, somewhere in your body, are going to be a ganglion. So the hard and fast rule between these 2 nervous systems is the fact that the pre-ganglionic neuron is found somewhere in the central nervous system. Somewhere in the brain or spinal cord, you find the pre-ganglionic neuron. And somewhere out there peripherally, you find the post-ganglionic neuron. 2 neurons in the chain, pre and post. So for parasympathetics, where do we find the pre-ganglionic neurons? Well, 4 of them are associated with cranial nerves. And guess what? They're located in the brainstem, close to where these cranial nerves actually originate from. So we actually have these pre-ganglionic parasympathetic neurons. Now, yesterday I mentioned this concept of ganglia. We talked about sensory ganglia. And I told you how the sensory ganglia have these, which you hadn't done with Dr. Cooper, which I was kind of surprised about. How you have these pseudo-unipolar neurons. These are only sensory. And even though we're using the word ganglion, in these autonomic ganglion, you find the multipolar neurons. So, multiple dendrites, 1 axon. Multipolar neurons have multiple dendrites, 1 axon. The pseudo-unipolar neuron only has the 1 process that comes off it. These are always sensory. And because autonomics are about motor, they cannot be these neurons. So I just want to make this clear because we are talking about ganglia. All right. So our preganglionic parasympathetic neurons are located somewhere in the brainstem for the cranial nerves. And then there's another component way down here in the sacral region that also has preganglionic parasympathetic neurons. So these neurons, which look like this, send their axons out of the central nervous system to find what? To find a postganglionic neuron out there somewhere. And the rule tends to be the fact that these postganglionic parasympathetic neurons are found in or near the end organs. So they're found near or in the end organs. So what does that mean? So you've already dissected the human heart, right? Or you've looked at models of the human heart. Did you actually get to dissect one? No. You didn't get a body. Oh, that's too bad. Well, anyway, when the day comes and you look at a human heart, you realize there's a lot of fat around it. And actually embedded in that fat of the human heart actually are little nodes of these parasympathetic neurons. So that's a really good example of these neurons being like literally in or very near the end organ. All right. So these preganglionic parasympathetic neurons located either in the brainstem or down here in the sacral region send their axons out. And their axons, as a very hard and fast rule, are always myelinated. So when they leave the central nervous system, they're myelinated by Schwann cells. And the axons during development sought out where the ganglia are located, and then they make synapses with these multipolar neurons found within those ganglia. Not ones that look like this, ones that look like this. Those postganglionic parasympathetic neurons and send their axons out. Their axons, because they're in or near the organ, tend to be relatively shorter than the preganglionics, and they're always unmyelinated. Now, this idea of myelination and unmyelination is not as critical here for parasympathetics as we're going to see it is for the sympathetics. Okay? But if the rule is, if you know that sympathetics are, the preganglionics are myelinated, postganglionics are unmyelinated, it equally applies to parasympathetics. So that's something that they have in common. This idea of myelination and non-myelination of the pre- and postganglionic axons, respectively. So, what are the four cranial nerves? That is absolutely critical to know. Oculomotor, facial, glossopharyngeus, and vagus. These are the four cranial nerves that carry preganglionic parasympathetic axons in them. These nerves down here in the sacral region contribute to the sacral plexus that's going to the urogenital system. So the sacral plexus going to the urogenital system. And we know by the cranial nerve names where they're going, right? Oculomotor, something having to do with the movement of the eye. Facial expression. Glossopharyngeus, things having to do with the pharynx. And vagus being the wanderer. Now, nowhere in there do we actually have the function of the parasympathetics, so you actually have to just know those. So what do these parasympathetics do collectively? Well, there's a list of them. So here are some functions. They cause pupil dilation. So of all of those pathways up there, right, starting at the bottom sacral, working our way up, which of those nerves would have anything to do with the eye, right? Pupil constriction. Well, that's oculomotor. Bronchioconstriction, cardiac deceleration. Well, vagus was the wanderer, right, that actually goes out of the head and neck and down into the abdominal and thoracic cavities. So that's got to be the vagus nerve, having to do with the heart and the lungs. Stimulation of digestion, vagus again. But salivation and lacrimation, well, that was facial and glossopharyngeus, as we're going to see again. And then some vasodilation going on. And the idea here is that, you know, the parasympathetic nervous system kicks in when the organism, the mammal, the human being, is at rest. So your pupils can be constricted, you know, you're actually kind of, you're sort of slowing down, you know, you're not really doing that much, and as you're at rest, you can actually digest the food you've eaten, and that's kind of where the parasympathetic nervous system kicks in. Now, this is a fantastic schematic that at times provide you with way too much information, but it does provide us with a lot of important things about these 4 cranial nerves and their parasympathetic component. Now, yes, you will have patients at some point in time who have problems with salivation. That's very true. And you certainly have to understand about the vagus nerve and what it does with regard to the heart and the lungs. Absolutely. But the first one at the top of our list is oculomotor, pupillary constriction. And it's absolutely important that you recognize all of the components of this nerve. So it turns out that if we think that it's a 2-neuron chain, the first neuron, when I say neuron singularly, you know that I mean a collection of neurons, right? There's a whole bunch of them, right? There's a whole bunch of these cells. It's not just one cell, right? There's a whole nucleus of these things in the brainstem. The first one is located somewhere in the brainstem. And for most of these, they actually get names. Now, the other ones, I'm not so concerned that you know exactly what those names are. But for scholarship, and because pupillary constriction is so important, it's not a bad idea to know that the preganglionic parasympathetic neurons are referred to as Edinger-Westphal nucleus. Now, it's an important concept about these sympathetics and parasympathetics to get, and might as well share it now. So if I were to show you a cross-sectional view of the midbrain, and I'm going to draw one on either side, big ocular motor neuron. So this is our cranial neuron number 3. These are the ones going out to innervate 4 of the 6 extraocular muscles plus levator palpebrae superioris. We know there's another nucleus called this Edinger-Westphal, and we know that those preganglionic parasympathetic fibers are associated with ocular motor. So where would this nucleus be? What would make the most sense? Well, it makes the most sense that it's actually immediately adjacent to this. And in fact, these preganglionic parasympathetic neurons are located immediately adjacent to the ocular motor nucleus, Edinger-Westphal nucleus. And the axons travel along with ocular motor. Now, these preganglionic parasympathetic axons are traveling along with ocular motor. And when ocular motor is damaged, you'll get both the loss of function of ocular motor to the 4 of the 6 extraocular muscles, plus levator palpebrae superioris, and deficits associated with the parasympathetic nervous system. But we know that these axons are going to have to leave this nerve. They're going to jump off because we have to find that postganglionic parasympathetic population of neurons. And they're located in a little tiny ganglion with a great name because it's exactly what they do, the ciliary ganglion. And if you think about what these fibers do with the ciliary muscles, right, and the pupillary constrictors, it kind of makes sense. So in the orbit is a tiny little ganglion. And as the ocular motor neuron passes into the orbit, these preganglionic parasympathetic axons come off. Because they're not going to do anything with the extraocular muscles, right? They need to find postganglionic parasympathetic neurons. So we have 2 parts here. The first neurons located in the brainstem, Edinger-Westphal, they travel out with the 3rd cranial nerve, but when they enter the orbit, they jump off. And when I say jump off, I'm not talking about a dynamic process happening all the time, right? Embryologically, they develop that way, and now we're just hardwired. But they branch off and they innervate the neurons in this little ganglia, and it's these neurons, these postganglionic sympathetic neurons, that innervate the pupillary constrictors and the ciliary muscles. And so both those are there on the schematic. So you've got everything about 3. You've got the name of the nucleus in the brainstem. You've got the cranial nerve they travel with. You've got the name of the ganglion they innervate. And you've got their target tissues in the eye that they innervate. The postganglionic fibers innervate. And that's great. Now, you can do the exact same thing with the other 3 nerves. It's just the fact that I'm not so hung up that you know about the inferior and superior salivatory nuclei or all these other things. But if we just look at it generally, we know that it's 7, 9, and 10. And if we think that what really is left with the smooth muscle component of the head, there's really nothing left except for glands. There's really nothing left. So we have our glands that need to be innervated. And our glands are the lacrimal gland, the large parotid gland, the submandibular and sublingual glands. And if you look, and through the chart I talked about yesterday and functions, it's facial that's responsible for innervating, sending axons that ultimately go to lacrimal, right, and sublingual and submandibular. And it's glossopharyngeus that carries fibers that ultimately go to or affect the parotid gland. And just like the third nerve, these preganglionic parasympathetic fibers actually have their own specific ganglia that have names. There's sphenopalatine, submandibular, which is kind of cool, submandibular gland, submandibular ganglion, and this little otic ganglion. You know, from a scholarship point of view, I think that would be great if you knew all those. But, you know, it's really not that imperative. What's so important is that you recognize the fact that there are 4 cranial nerves that carry preganglionic parasympathetic fibers. So why do I say preganglionic? Because eventually those preganglionic fibers have to jump off that cranial nerve and find their ganglia. And it's the fibers from the neurons in the ganglia that innervate the target tissue, like the parotid, sublingual, submandibular, and lacrimal glands. Vagus, our 10, is leaving the head and neck, and it's going south. And it's going to the lungs and the heart and the upper part of the gastrointestinal tract. So 3 is absolutely fair game to ask you anything about. It's so clinically relevant. You should know all the components. I don't know who Edinger Westphal were. I don't know what else they did from a neuroscience point of view other than find these neurons. Having said that, though, that's the name it's called, and that's what you should recognize. But I want you to have an understanding about this concept, that the neurons that affect the extraocular muscles associated with cranial nerve 3, those neurons are different from the parasympathetic neurons, the preganglionic parasympathetics. And what those preganglionic parasympathetic axons do is, basically, since you're going in the same direction, I'm just going to follow along for a while. Makes sense. Ocular motor is going to the orbit. These preganglionic parasympathetics have got to go to the orbit. So they travel along, and then when they need, they leave that nerve to then find that postganglionic population of parasympathetic neurons. Yeah?
So 3 doesn't have actual parasympathetic effect?
Absolutely. So every time that you'll meet a patient with a third nerve palsy, it's the third nerve is affected when those parasympathetic fibers are still there. So we're going to talk about that later. So you look at the cranial nerve here. You think about where the third cranial nerve is coming out of the brainstem and traveling before it enters into the superior orbital fissure to enter into the orbit. This distance here is the vulnerable distance. This is the area where 3 is going to be damaged. If it's damaged in here, let me tell you, there's a lot more going on, right? Because that's trauma into the orbit. The eye's going to be damaged, the muscles are going to be damaged. Like that's completely different. Third nerve palsy, third nerve injuries occur out here, between the brainstem and the superior orbital fissure. And it's in here that 3 has the axons for the 4 of the 6 extraocular muscles. It has the axons for levator palpebrae superioris, and it carries the preganglionic parasympathetic axons for pupillary constriction and near accommodation. So what does that mean? Neurons work because they're influenced by other neurons. So, let me draw this thing. a fundamental core concept in neurosciences. So, here is our Edinger-Westphal nucleus neurons, our preganglionic parasympathetic neurons. They travel in the third cranial nerve. They innervate other neurons in the ciliary ganglion. And it's these neurons that then innervate the pupillary constrictors. Three is vulnerable here, where the axons have left the brainstem and between the large opening here, the superior orbital fissure. This is where it's vulnerable. Which means the fact that if there was a space-occupying lesion, like a tumor growing, right, adjacent to 3 and pinching it off, these axons don't work. These neurons are still functional, but these neurons are relying on information from 3, the preganglionics. So neurons rely on each other to tell them what to do. They don't work autonomously. So these postganglionic neurons only work when they're receiving signals from the preganglionics. If you damage these axons in any way, that's where you will see a dilated pupil, because the constrictors are not working. Okay? So this concept of neurons influencing each other it's so inherent to the way I think, and I just want to make sure I get that concept across, because I think that's kind of where your question was based. Yeah. So, even though this neuron is not damaged, right, and it's still innervating the target tissues, it relies completely, 100%, on the information from this neuron here. And as we're going to these from sympathetics, they even rely on information from the next neuron up. So it's kind of cool. So neurons are always talking to, they're always this chain of events of signals. And so yes, absolutely. So the vulnerable zone for 3 is here, between the brainstem and the superior orbital fissure. There is unlikely to be a very precise and specific damage only to the ciliary ganglion, only to these postganglionic parasympathetic axons. It's never going to occur. It would occur with traumatic injury of the eye. But then, of course, the eye would be damaged, the muscles would be damaged, the fibers and the ganglion would be damaged as well, because it's just a delicate little ganglion inside the orbit. And again, just to emphasize this concept one more time, is the fact that when we talk about these cranial nerves, everything is completely ipsilateral. So left ocular motor, right, is affecting the left eye, right, carrying the constrictors for the left eye, and vice versa for the right. So absolutely critically, you recognize that. All right. So that's parasympathetics. Now, back in the brain, back in the sacral region, down here, way down here, one thing to mention is how do these axons exit out of the spinal cord? Because now it's not the brain stem, right? Now we actually have the spinal cord. And remember I said the spinal cord is segregated into 2 halves. The upper half, or dorsal part, is all sensory. And the lower ventral half is all motor. And if you think about what these axons do, they're motor to smooth muscle, then they should exit out that ventral root. Makes sense, because they are in fact a type of motor neuron. And their axons do in fact exit out the ventral root. They pass in the spinal nerve, they contribute to some sort of plexus, and then find a ganglion out here that's going to then go on to affect the urogenital tract in some capacity. So there's nothing associated with the dorsal root. That's all sensory. This is motor. And so the cell body, again, the first one, the preganglionic neuron, is located centrally, somewhere in the central nervous system. Now this is between sacral levels 2 and 4. It exits out the ventral root, comes into the spinal nerve, and again it finds that postganglionic sympathetic neuron, postganglionic parasympathetic population. somewhere near or within the end organ, like the urinary bladder or the reproductive organs. Okay, so sympathetics. How do we deal with this one? So why don't we do the similarities first? How are parasympathetics and sympathetics similar? So similarity number one, it's this two-neuron chain. The preganglionic neuron found in the central nervous system, postganglionic neuron found peripherally. Is that true? Yeah. The preganglionic sympathetics are found in the spinal cord, and the postganglionics are found out here in a series of different types of ganglia. Same thing. The preganglionic axons are myelinated. The postganglionic axons are not myelinated. Same for both. And that's pretty well where the similarities end. The first major difference is where do we find these preganglionic sympathetic neurons? And if you look at it, it's completely different from the parasympathetics. Now there's nothing in the brainstem, there's nothing in the sacral region. Rather, these preganglionic sympathetics are found between thoracic level 1 and lumbar level 3. Or 3. The ganglion, where we're going to find those postganglionic sympathetic neurons, have to be different as well. We're talking about a completely different set of ganglia. But within those ganglia, again, are those multipolar neurons, not the pseudo-unipolar neurons. These are motor neurons. And then the action of the sympathetics is, again, completely different. Now we get this idea of fight or flight. So if you're going to stand and fight, you need to have pupillary dilation. Your eyes need to take in all the visual information you want. You want to get more oxygen into the body, so bronchial dilation. You want to get the heart going. You don't have any time now for digestion. Think of, two cats fighting, piloerection, right? All the hair standing on end, right? And systemic vasoconstriction, well, you want to cut off the blood supply internally and shunt all the blood supply, to your skeletal muscles. So that's actually collectively what they do. But look again who's first on top of the list here, the pupil. So parasympathetics are pupillary constriction, sympathetics are pupillary dilation. So what we need to figure out now is how the sympathetic nervous system is organized. How does it actually work from an anatomical point of view? So it's here where you really get the idea of this business of the axons being myelinated or not myelinated. So since you have not done any cadaveric dissection, correct? Is that a yes? You have not done it. You have not done it. Correct. Oh, you have done certain parts. Did you do the thoracic cavity? Did Dr. Coe ever show you where the sympathetic chain was? Does that ring a bell? All right. So, the thoracic cavity, when you look in the back of it, there is a chain that goes all the way down, and there's some really nice representations in your gross anatomy text. You should have a look. And this chain is referred to as the sympathetic chain. And the sympathetic chain, by its very name, is one of those ganglia, where we're going to find the postganglionic sympathetic neurons. Now, what the artist is doing here is representing the spinal cord and the sympathetic chain only on one side. So the exact same thing is happening on the other side, it's just not represented. So it's just making it simpler for us to understand. So we have this sympathetic chain that contains these postganglionic sympathetic neurons. But in fact, there are other ones there are other ganglion out there that are associated with the major blood vessels of the gastrointestinal tract. So have you done your anatomy of the GI tract? Good. So if I were to give you three names, celiac, superior mesenteric, and inferior mesenteric, what word would you put behind it now? Artery, right? Celiac artery, superior mesenteric artery, inferior mesenteric artery. Well, you can add to that now ganglia, because these prevertebral ganglia sit immediately adjacent to those major blood vessels. And we're going to see why in just a moment. All right. So we've got several sympathetic ganglia out there. We've got a big chain, or we've got these other ganglia. So paravertebral, the way I always remember it, para kind of next to, so it's next to the vertebrae, and prevertebral means before the vertebrae, it's somewhere out there, before we get even close to the vertebral column. So these, and I've got it written down that the prevertebral ganglia include celiac superior and inferior mesenterics. Okay. Now what I need to do next is explain to you the different possible routes. Now, I've written it out for you. So it's best, I think, to go through it visually, but all the information is written out clearly here. So I'm just going to iterate what I've written, okay? So we have to think that where are these axons going, what are they doing, how are they influencing, and organs. So we know that the preganglionic sympathetics are found between thoracic level 1, lumbar level 3. We find them in a very specific nucleus with this kind of long name, but it's intermediolateral. So it's kind of intermediate in the medial aspect, you know, on the lateral side of the spinal cord. It's kind of a silly name. But nonetheless, it's located where? In the dorsal half or the ventral half? It's got to be ventral half, right? I mean, because these, in fact, are motor fibers. They're influencing motor activity. So these preganglionic sympathetic axons leave the spinal cord via the ventral root. They join the spinal nerve. Now, the way the sympathetic chain is attached to the spinal nerve are by these two structures referred to as either the gray or the white ramus. And the words gray and white refer to exactly the same way here as they do when you describe the brain, the gray and the white matter. The white are the myelinated structures, and the gray are the ones that are not myelinated, which makes them have a more dull appearance by comparison. So gray, which really means if I look at it and I just go by that simple definition, I know that the white ramus, or the little connection between the spinal nerve and the ganglia, must have myelinated axons. Well, go back to the rule. All preganglionic autonomic axons are myelinated. And in fact, it's still the first axon. So the neuron located in the spinal cord sends its axon out to the ventral root, enters the spinal nerve, and loops around to the white ramus. White because they're myelinated. The first option as to find that postganglionic neuron is to find a neuron at the same level between the sympathetic chain, which is extending all the way on either side of the vertebral column. So to find a neuron right around the same level as you would find between the pre and the postganglionic neuron. And that preganglionic axon synapses, forms a contact with that postganglionic neuron, and then it sends its axons out there peripherally. So that's option one. And as it sends its axons out, it grows back into the gray ramus. Gray because these axons are unmyelinated. And out they go. So this is really where myelination is somewhat important, right? It gives you this idea of the entry and the exit through the sympathetic chain. So, option one is this. The preganglionic sympathetic neurons send their axons out to the ventral root, enter the spinal cord, white ramus, find a neuron somewhere around the same level between the sympathetic chain and the spinal cord. That postganglionic sympathetic neuron sends its axon out to the gray ramus and into the spinal nerves to do all sorts of things, which we'll talk about in just a second. All right, option one. Option two. Because the sympathetic chain is a collection of neurons on either side of the vertebral column, option two is the fact that the axon can enter the sympathetic chain, but go up or go down to find a neuron at a completely different level. And when it does that, it just finds a neuron at another level, and then it again sends its axon to the gray ramus, and on we go to innervate target tissues. All right. The third option is a little different. The preganglionic sympathetic axon has to enter the spinal nerve through the white radius, but if you look at it, it just passes right through the sympathetic chain and finds these these postganglionic sympathetic neurons in the prevertebral ganglia. Then they go off and innervate in tissues some distance away. And the last one I have there is sort of this exception to all of the rules, which is the fact that the preganglionic sympathetic neurons actually directly innervate adrenal medullary cells, which are considered to be modified postganglionic neurons. All right, what does all this mean? So if we put it all together in, again, what I think is a great schematic, it's all here. So let me just go through it all from the top and have a nice review here. So if we look on the right side, we've got our parasympathetic component. How do we know it's parasympathetic? Because we've got our 4 cranial nerves up here and our sacral division down here. That's parasympathetic. And here in the middle, we actually have all the organs that are going to receive this dual innervation. And look who's at the top of the list again, the pupil. Pupillary constriction, pupillary dilation. And all the other organs, yes, they are important, but definitely the pupil's at the top of the list. And on the left side here, we actually have the sympathetic nervous system, because here's our sympathetic chain, very long and extensive. And again, remember, everything is bilateral. It's just showing you only on the one side, okay? And our preganglionic sympathetics located between T1 and L3. So, if we look at something like pupillary dilation, let's start with the pupil, and pupillary constriction. How do these preganglionic axons and these postganglionic neurons work? So for pupillary constriction, those preganglionic parasympathetic neurons originate in the Edinger-Westphal nucleus. They project out an ocular motor nerve, and they find those postganglionic neurons in the little ciliary ganglia, and those postganglionic sympathetics go off to innervate the pupillary constrictors. But the balance is the sympathetics. And they're a great example of one of these preganglionic sympathetics sending their axons out into the sympathetic chain, and then ascending to find another structure. And that structure is the superior cervical ganglion. This ganglion sits at the bifurcation of the carotids. So that superior cervical ganglia then sends its axons up to actually cause pupillary dilation. How do you think these postganglionic sympathetic axons get from here in your neck at the bifurcation of the carotids, from here to the pupil to cause pupillary dilation? What would be the easiest way? They could create their own nerve. Absolutely, they could, but they don't. Sympathetics, postganglionic sympathetics, love to travel along blood vessels. In fact, every artery in your body is absolutely covered in postganglionic sympathetic axons. So they get from here to the pupil. Why not travel along the carotid? Because it's kind of going in the same direction. And if you think about the vascular lecture that Dr. Cooper would have given you, right? So you can those axons can travel along the internal carotid artery, and the first major branch off the internal carotid is actually, once it enters into the cranial bulb, is actually ophthalmic. And since ophthalmic is going to the eye, why not just have the sympathetics travel along the ophthalmic artery to get to the eye, and then again jump off the blood vessel to actually innervate the pupillary dilators? So when you think about dilation and constriction of the pupil, one of them, this part here, is completely all about nerves, real nerves you can see, ocular motor, and these much shorter parasympathetic fibers. Whereas for the sympathetics, these postganglionic sympathetic axons, these unmyelinated fibers, love to travel along blood vessels. And they do that by traveling along the internal carotid and then ophthalmic to actually get to the eye. So we can see then there's also the balance with regard to the salivary glands. Oh, that's not a big deal. And then we actually get to the heart, and you can see how vagus is contributing to the parasympathetic innervation of the heart. And we have these upper thoracics doing the heart. And you can go through all these tissues right down the list. There are, however, a couple of tissues on the schematic that are not receiving a dual innervation. And if you look carefully, there are the sweat glands, there are piloerection, getting your hair to stand on end, and your blood vessels. And if we just said how sympathetics love to travel along blood vessels, then it makes sense the fact that they're responsible for affecting the blood vessels themselves. And so when you think about sweat glands, when you think about piloerection, and you actually think about blood vessel constriction, it's just the sympathetics being on with no balance to parasympathetics. So sweating is a sympathetic function. No parasympathetics shutting off. Either the sympathetics are on or they're not. Same for piloerection. So the last thing I just want to mention is, again, just to iterate about these ganglia. So if I were to say now that we have these sympathetic ganglia here at celiac, superior mesenteric, and inferior mesenteric, and we want these postganglionic sympathetic axons to get from the ganglion to the stomach, or from the ganglion to the intestines, or from the ganglion to the urinary bladder, what would be the easiest route for these postganglionic sympathetic fibers to take? Along the blood vessel. The blood vessel is going in the exact same direction, right? So if you think about the celiac artery supplying blood to the stomach, superior mesenteric supplying blood to the intestines, inferior mesenteric supplying blood to the bladder, just follow along the blood vessel. And then when you get to the end organ, you just jump off to innervate those smooth muscles. So sympathetics love to travel along blood vessels, arteries, not so much veins, but definitely arteries. So everything is there on that schematic. And as I said, there are lots of schematics out there, all sorts of ways of representing this, but everything is nicely organized on here. And again, if I was actually going to do it from a learning point of view as to what you want to make sure you know, start at the top of the list. So when you're shining light into people's eyes and looking to see if they dilate or they do not dilate, they constrict or they're dilating or whatever they're doing, that you understand how the pupil functions in human beings. So this is a review of your cranial nerves and a list of which ones have parasympathetic function. That's just somatic, which we're going to do right now. And then I'm going to start here. So here is our superior cervical ganglion. And this is actually, you know, I don't want it to seem like I'm really, you know, hammering a point home. But I really do think, you know, for your own scholarship, that, you know, from a clinical perspective, you have to understand how the pupil works. And you have to understand which neurons are associated with the functioning of the pupil. So we know that oculomotor is the one bringing the parasympathetic fibers. But I want you to be clear about how the sympathetic part is working, because there is one condition that actually sort of provides people with a little confusion. So let's go and just ask ourselves about the two basic neurons, pre- and postganglionic sympathetic fibers. And if we're talking about the eye, and if you think about that huge chain of preganglionic neurons in the spinal cord, it makes sense that the eye would be influenced by the neurons at the very top of that chain of neurons in the spinal cord, and that's around T1, T2. So those preganglionic sympathetic neurons send their axons out. They go to the white ramus, and they ascend in the sympathetic trunk to find our superior cervical ganglion at the bifurcation of the carotids. Rather than creating their own nerve, they travel along the internal carotid, and what's not shown here, but they travel then along ophthalmic to reach the eye, where they will actually cause pupillary dilation. Now, that's absolutely great. Now, what if the postganglionic sympathetic neurons were not working? What would you expect to see? If the postganglionic sympathetic neurons, let's say there was some sort of tumor growing in the neck, a metastatic tumor that was infiltrating this ganglion and causing tremendous disruption, right? Then you would expect on this side to see a constricted pupil. The balance now is gone, right? These neurons are not working, the balance is gone, it's all ocular motor causing constriction. That's easy. What if these neurons are fine, but the lesion now is in the spinal cord or in the exit route for these preganglionic sympathetics? Again, remembering how they influence each other, even though these are intact, these are affected, the system doesn't work, and the person would have a constricted pupil. That's great. But even these preganglionic sympathetic neurons are influenced by other neurons. And these other neurons are way up in the hypothalamus. And these hypothalamic axons come down, and they innervate these preganglionic sympathetic neurons. They're not sympathetic, they're now hypothalamic. And, they're regulatory. If there's now a lesion in the brainstem, not in the spinal cord, not in the superior cervical ganglia, but in the brainstem, cutting off those regulatory hypothalamic projections, guess what? You get a constricted pupil. So it really shows you how these hypothalamic neurons are driving these spinal neurons, and how these spinal neurons are driving the ganglionic neurons, and the ganglionic neurons are the one causing pupillary constriction, pupillary dilation, ending up with a constricted pupil. So neurons are all talking, they're all sharing information, okay? So, and the reason why I put it up there is because people think somehow that the sympathetics are traveling in the brainstem by these brainstem lesions, that you actually end up with a sympathetic sign. Couldn't be further from the truth. And that's what this schematic is so nice. So it actually shows you the fact that a lesion in the brainstem can actually give you a sympathetic sign that something's wrong with the sympathetic nervous system. But the spinal neurons are intact, the ganglionic neurons are intact. What you disrupted is the regulation of the activity. And that will cause to have a constricted pupil because the parasympathetics are working just fine. All right, let's take a break, and I'll tell you one last thing about this stuff, and then we'll start motor systems. So, one other thing to mention here about these sympathetic fibers that travel along the internal carotid artery and ophthalmic to end up going to the orbit, where they innervate definitely the pupillary dilators. They also innervate a set of muscles in the upper eyelid. We've already done a set of muscles in the upper eyelid, levator palpebrae superioris. And it turns out that we have another set of muscles in the upper eyelid referred to as the tarsal or Mueller's muscles. Has anybody mentioned those to you before? No? All right. So, if I were to tell you, and now which I have told you, and now that you already know, that levator palpebrae superioris is innervated by ocular motor, right? Ocular motor, the somatic part of ocular motor, then these muscle fibers have got to be what type? Skeletal. If I tell you that these tarsal muscles are innervated by postganglionic sympathetic fibers, they have to be smooth, right? They can't be cardiac, right? All that's left is smooth, right? So for human beings, actually for all mammalian species, because you can actually do ganglionectomies in mice and get little droopy eyelids, a droopy eyelid is referred to as ptosis. P-T-O-S-I-S, ptosis. And it turns out that for us to keep our eyelids just up, without really consciously aware of keeping them up, right, that both sets of muscles have to work. What does that mean? That means that a lesion of ocular motor one of the signs is ptosis, a droopy eyelid. Because if ocular motor is not working, then the levator palpebrae superioris muscle is not working, and the patient has a droopy eyelid on the affected side. But it turns out that if the sympathetics are not working properly, you'll also get a droopy eyelid on the affected side. So our upper eyelid requires both sets of muscles to be working and functionally innervated. And so if you interrupt either, you'll get a droopy eyelid. But what's the big difference with regard to the pupil? So if ocular motor is not working and you have a droopy eyelid, would the pupil be dilated or constricted? It would be dilated, right? Because ocular motor is not working, therefore constriction. Therefore, you have the unopposed action of sympathetics. You'd have a droopy eyelid and a dilated pupil. And as well, the eye would actually be moved away, but we're not going to work. So just a droopy eyelid and a dilated pupil. If the sympathetics aren't working, you're going to have a droopy eyelid, but now a constricted pupil. Because the sympathetics are responsible for dilation. If they're not working, you have the unopposed action of ocular motor, and then you have a constricted pupil. So ptosis is great to know that something is wrong, but it really doesn't help you as knowing whether it's sympathetic or parasympathetic. And that's really where the pupil comes in as to its natural state of what's going on. Does it appear constricted or dilated compared to the unaffected side? Now, one little note I'm just going to make here, and only because we will be talking about sensory systems, but not visceral sensory systems. If you think about it, all of these things have to do with visceral motor activity, right? The heart, the lungs, the digestive system, the urinary bladder, etc. And as much as you need motor activity to drive those systems, what about sensory activity coming back? And so there are, in fact, all these tissues do, in fact, have sensory fibers in them. I mean, you clearly know when your bowel is distended. You clearly become consciously aware when your urinary bladder is distended, right? That the conscious necessity to void. And so these are sensory fibers coming back. And when we think about these sensory fibers, don't forget that they're these pseudo-unipolar neurons. Their cell body is situated in that dorsal root ganglia, and they have to share again the information centrally. And that idea of sharing information, who are they going to share it with? Well, they're going to share it with these autonomic neurons, the sacral parasympathetic neurons or the thoracic lumbar sympathetic neurons. But they have to share that sensory information with someone. So we get all sorts of things about stretch in the bowel and the urinary bladder, lack of oxygen definitely in the heart. When a heart attack is going on, lots of sensory activity then. But just don't forget that these autonomics are all about motor. But these tissues we're talking about definitely have sensory innervation. And for the most part, they're silent, right? Don't really know there's anything wrong right now with many of my organs, right? But you certainly know when your bladder is distended, you need to void. You certainly become aware of your bowel when things are not going well, right? And for the most part, you never hear anything from the heart, which is a really good thing, right? Lack of oxygen can't be particularly very good, okay? So just don't forget the fact that these autonomics are about motor, but there are these sensory axons, and they would be completely different. These are those pseudo-unipolar neurons where you would find their cell bodies in the dorsal root ganglia. And that's just the schematic again. Great. So now we've done the involuntary motor activity. So now it's time to start thinking about voluntary motor activity. So, what do we need to do? We definitely need to figure out these two terms, upper and lower motor neurons, because I know for a fact that when you go to the clinic, you are going to be exposed to senior residents and doctors talking about a patient having an upper versus a lower motor neuron disease. And you have to understand what those terms mean. We have to figure out about the different pathways, and the one that is really the major workhorse of this voluntary motor activity in our body is the corticospinal pathway. We have to understand a little bit about the other helper pathways like rubrospinal, vestibulospinal, and definitely we have to talk about corticobulbar, but that's going to be left for the brainstem lecture. But it's absolutely very critical to make sure that I mention that as well. So, what are we talking about? We're talking about voluntary motor activity. That you give someone a command to pick up the marker, and that they voluntarily extend their hand forward and they grab the marker with the right amount of pressure, the right amount of force to pick it up and hold it in their hand. That's voluntary. So how do we do it? How do we actually get that wonderful fluid motion, right? Smooth motion of the arm that goes forward. And even just by me looking at the object, I have a good idea about how much force I need and how much it probably weighs that I will be exerting to actually pick it up and to hold it in my hand, right? So this wonderfully controlled motor activity really is in the cerebral cortex. And so this schematic here represents how we get this flow of activity from the cortex down to the spinal cord to actually allow us to do very precise things with our hands and with our limbs. Now, again, this concept, and I just want to make sure that you get it, is the fact that neurons need to get signals from other neurons. Like, there's no autonomy here. And so we can look at the cerebral cortex and say, well, yeah, it's great. That regulates motor activity. But it, in fact, is regulated. And it's regulated by structures we haven't done yet, like the thalamus. And there are even other structures that I've mentioned in passing, the pons and the cerebellum, the brainstem, right? And this other structure referred to as the basal ganglia. Now, this is always an exception to the rule in science, I find. And this is that one exception to the rule of terminology. So I've told you that ganglia is a collection of neurons outside of the central nervous system. Unfortunately, somewhere a long time ago, this term basal ganglia applies to a group of neurons in the central nervous system, and it has stuck. Can't do anything about it. That's just what it's called. But anyway, so we have all of these structures which collectively are influencing the activity of the cerebral cortex so that we get that wonderful activity, that outflow of motor activity that is precise and is directed. Okay? Now, we're going to actually talk about all these other different components, basal ganglia, cerebellum, pons, thalamus, brainstem, but today it's just to actually get through the voluntary motor activity. So what is the actual pathway that's going on here? So when you give a patient a command to wiggle their toes on their right foot, let's get beyond the comprehension of the language. Let's get beyond the comprehension of what part of the body you want them to activate. But once that signal is there, you don't have an individual wiggling their left fingers, right? The patient wiggles their right toes. It's clear. So we need to activate those cortical neurons that specifically drive the activity of the toes on the one foot. And so the nervous system has got to be set up with this tremendous degree of specificity. That when you ask them to smile, someone smiles. When you ask them to move their arm in a sort of a nose to finger fashion, they're doing exactly what you're asking them to do. They understand the task and they do it. What you're assessing is whether or not they can do it and how well they do it, right? So there has to be tremendous specificity here, that you have populations of neurons that are dedicated to activating specific populations of digits, of muscle groups within your body. So we actually have a 2-neuron chain, and it could not be simpler. Upper and lower motor neurons. The upper motor neuron is located in this structure called the primary motor cortex. Now, this gyri is a deep groove in the cerebral cortex, and in front of it is where we actually find the primary motor cortex. This idea of a precentral gyrus is actually where we find the primary motor cortex. So, again, remembering the fact that dot represents the neuron cell body, the line represents the axon, right? And we have an upper and a lower. So the uppers are located in the cerebral cortex. And not anywhere in the cerebral cortex, but specifically in this primary motor cortex that's found in the precentral gyrus. These neurons have to be organized in a very topographical manner that represent the body. Because the voluntary information you're giving, you want to activate that specific population of neurons. So when you ask the person to wiggle their right toes, they don't start, wiggling their left fingers. They know exactly what's going on. So remembering the fact that all the schematics we're looking at are showing one side, not both. So we know that there is a primary motor cortex on the left hemisphere here and on the right. Our axon has to get from the cortex down to the spinal cord. Because you know the fact that to move your hands, right, the fingers of your hands, the toes of your feet, that's a spinal activity. That there are neurons in the spinal cord that extend their axons out to innervate skeletal muscle. And it's skeletal muscle because this is all voluntary activity. So here are our 2 neurons. One's in the cortex, specifically located in the primary motor cortex, and the other one is located somewhere in the spinal cord. Because the neurons to move your hand must be very different from the neurons that move your foot. And obviously your hand is up here in the cervical region, and your foot is going to be down here in the lumbar region. But you have motor neurons all the way throughout. Now, when you actually look at the clinical signs of upper versus lower motor neuron diseases, look collectively again what's happening. If you think that you need this neuron to send a signal, neuron 1 to send a signal to neuron 2 to get the voluntary motor activity to work, that means again, if you have a lesion in the upper motor neuron, a lesion of this neuron up here, you would end up having weakness in the muscle. The muscle's not going to work. It's not getting the proper signals. Even though the lower motor neuron is fine, right? It's just not receiving the appropriate signals. So you would get weakness for both. It turns out, though, again, it's this wonderful dependence between skeletal muscle and their axons that innervate them to actually sustain the shape of it and the appearance of it. Remember I told you about the hypoglossal nerve? Damage to it causes atrophy of the tongue on the one side. The muscles waste away. It's only because they don't receive that innervation anymore, right? Skeletal muscles love to work, and the way they work is by receiving innervation by nerves. Atrophy definitely occurs when the lower motor neuron, this one down here, is not working. It's not sending the appropriate signals. It's not even present within the skeletal muscle, or it's not working. And there's this idea of trophic dependence between these 2 tissues. Fasciculations are where the muscle begins to twitch, and that definitely is something that occurs when the muscle is denervated, and not so much when there's an upper motor neuron disease. And the reflexes are definitely, and reflexes and tone are definitely something that Dr. Cooper will do with you again in your term 5 when he does neurology again with you. So we have 2 neurons, upper and lower motors. So let's look at the cortex. So here is that primary motor cortex. Here is this central sulcus, this very deep groove, and in front of it is this gyro. Now, this tissue is very specific. So in all of us, our primary motor cortex is here. It's not over here. It's not down here. And all of this, it's in exactly the same place. That's very important. We have one on the left and one on the right. So we're looking at a lateral view here. Now, these numbers refer to something called Brodmann's numbers. Brodmann was an incredible histologist, and he thought that the human cerebral cortex must have a degree of heterogeneity in it, just in the histological organization, because he knew the fact that this area here is your primary motor cortex. This is what drives voluntary motor activity. Not this area, not this area, not this one. And he wondered whether or not the functional differences between these areas of the cortex matched some sort of anatomical differences. And sure enough, he was right. That histologically, the organization of the neurons, the density of the neurons, the size of the neurons shifted in all these different parts of the cerebral cortex. And he could match them up precisely with function. So that was incredibly cool. That's, for me, to actually think is cool as a neuroanatomist. For you, what's important, unfortunately, is the fact that I know you will have a clinician of, there's somewhere who will say to you, area 4. And you'll think to yourself, area 4? What's area 4? Well, you know, you only have a few important ones to learn. Okay? And clearly, Brodmann's area 4, the primary motor cortex, is one to learn, commit to memory. Not the association motor cortex or the premotor cortex. Don't worry about their numbers. All right. So we have our primary motor cortex that is distinct functionally, distinct histologically from all the other tissues around it in the cerebral hemispheres. And I want you to recognize the fact that this lateral view here, our primary motor cortex extends all the way right from the big interhemispheric fissure between the two, the left and right hemispheres, extends all the way down here laterally in front of this very deep groove called the central sulcus. And in fact, if you look at the medial aspects, so we did the sagittal view, we did a cut here of this structure, and we actually look medially, we can still see some of the primary motor cortex right there. So these are both medial and lateral views, and the major thing is we want to make sure we understand this primary motor cortex. Now, if you just appreciate the fact that the neurons in here do not work by themselves, right? When you give the patient the instruction to wiggle their left toes, they need to understand that, and they need to funnel that information specifically to the motor cortex that has to do with the left toes. So there are all sorts of cortical areas that help with this decision making. But ultimately, the signals are going to come from that specific part of the primary motor cortex. So how do we determine this? This is just more about Brodmann. I'll come back to this. So this is a structure, the schematic is referred to as the homunculus, or the little human. So what is represented here? Well, what's represented are both the primary motor and the primary somatosensory cortices. And if we look top down, we can see here, if this is the nose in the front, and this is the back of the head here, right, that we have wonderful symmetry on left and right. We have a primary motor cortex and a primary somatosensory cortex. So let's just do the primary motor cortex, because that's what this lecture is about. You want the patient to wiggle their left toes. And I hope that you can appreciate the fact that the cortical tissue here is extensive. But within here are millions of neurons. All neurons in here, millions of them. And these millions of neurons have to be segregated. They have to be. Because you need neurons that are specifically going to affect the toes versus the hand, the knee versus the elbow, the hip versus the shoulder, and especially for the hand, for all the precise things that human beings do with their hand, and absolutely with the face, for all the precise things we do with our face, in particular, speech. So when you think about it, there is this regionalization of function. There has to be. So we have neurons in the primary motor cortex that start here medially that are about the foot. We go from the leg, the knee, the thigh, the trunk, the shoulder, the arm, the elbow, the forearm, the hand, and the face. And what this is really represented is not only the precise localization of these upper motor neurons. That's the first thing it's represented, is the precise localization of the upper motor neuron. But it's also representing the relative proportion of neurons you need to affect function. What does that mean? Well, the number of neurons you need to wiggle your toes are not nearly as many as the number of neurons you have dedicated to do this with your fingers. And that makes sense. Because what the human beings do with their fingers is a heck of a lot more important than what human beings do with their toes. And so the number of neurons you need for your hand is proportionally larger than the number of neurons you need for your foot. And then on this motor cortex, we can actually see that there's this huge part here of cortical tissue, a massive number of neurons that is required for the movement of the face. Now, not only when we think about movement of the face, but we also have to think about movement of what for the articulation of speech? The tongue, the pharynx, and the larynx, right? All of which are voluntary motor activity. And they're tucked right down here next to the face. So that's what's going on with this structure. It's a representation of the relative proportions of neurons needed to affect specific activity of those skeletal muscles. And it also shows you their precise localization within the cortex. So, and that's going to become very important when we think about regional lesions within the primary motor cortex. Now, for tomorrow, when we talk about somatosensory, if you just by comparison look, the homunculus is very similar. And so what is somatosensory? Somatosensory is, where is the sensory information coming from? So ask a patient to close their eyes, right? And you touch their left forehead versus their right cheek. They can describe to you exactly where the sensation is coming from. You touch their right hand, their left hand versus their right foot. They can describe it. How do they know where the information is coming from? It's the primary somatosensory cortex. We'll do that tomorrow. So today is about how to affect motor activity precisely. And that is what we're doing. Now, what we're going to do today is about the corticospinal and the spinal pathways. So today what we're just talking about is everything from the neck down. Okay? The neck down. I'll mention the neck up a couple of times, but for the most part, we're talking about corticospinal. Right. That's just so a little something about a little bit more about Brodmann, and just one of the interesting things is the fact that it's these large neurons here. Just in case this may come up to you later. So Brodmann actually defined 6 distinct parts, layers in the cerebral cortex, and I hope that even you can see from back there is the fact that these really large meganeurons located in layer 5. And they're only located in layer 5 in the primary motor cortex. And these large motor neurons are driving the precise movement of your hand. Right? That's in fact how important it is. And they make these monosynaptic connections that actually provide for very dexterous movement of the individual digits and joints, and predominantly of the hand. Okay, so now we know where the neurons are located. So now it's about how do we get these neurons, their axons, from point A to point B. So there are 2 schematics on here. One makes perfect sense, the one on the left, and the one on the right, it's like, what the heck is that? So I'm going to try to piece it together bit by bit. All right. So originating from our area 4, our primary motor cortex, is a huge bundle of axons, right? So now we have hardwired, you've got all these neurons innervating, all these motor neurons, and you're only going to activate the ones to actually move the left hand. So these axons descend through the brain in a structure referred to as the internal capsule. And the internal capsule just gets its name because these are myelinated fibers. Motor activity you want to occur very fast, right? So these are big axons that are myelinated. And it's just referred to as the internal capsule. And these axons just keep descending because we're actually heading toward the spinal cord. And there's actually a very important event that's just about to occur. Now, right off the bat, we have this distinction between the body, the neck down, and the head, the neck up. The cortical axons that are heading toward the spinal cord are referred to as corticospinal. So they're going to influence the body from the neck down. The cortical bulbar fibers are influencing everything having to do with the face, the neck up. So we get this distinction between corticospinal and cortical bulbar. And what's really cool here is the fact that this is actually a very odd sort of sectional view to the brain, very deep. All these things here we haven't done yet, okay? Don't worry about that. But what it's showing you is something really interesting about how the brain gets developed and how it's organized. And that's this. If you think about these two major parts of your body, the head and everything from the neck and below, you want to segregate that information right off the bat. You want to segregate these axons because they're going to two completely different places. And right off the bat, we've got cortical bulbar axons descending to the brainstem, right? Through to the brain and onto the brainstem. And then we have our cortical spinals. And the axons within the cortical spinal pathway are not all kind of higgledy-piggledy mixed up with each other. All the ones heading toward the upper spinal cord for the arm are traveling together. All the ones traveling together for the thoracic and upper lumbar, for the trunk, traveling together. And the ones having to do with the leg, lumbosacral, are traveling together. And that's what this ATL stands for. Arm, trunk, leg. The nervous system, when it's developing, parcellates all these things out. And it makes sense that they should all travel together because just like a highway, they're all going to sort of exit off at the same point. And so why not all travel together as you're descending through the brain? So right away we get a segregation between the neck up and the neck down. And even these corticospinals are segregated inside that pathway with regard to the axons going to the arm, trunk, and the leg. Now, as they descend, they're all traveling on the same side. So we've got both a corticospinal pathway on the left and a corticospinal pathway on the right. They're all traveling down through the brainstem, and they get to this really interesting area called the cervicomedullary junction. The cervicomedullary junction just refers to the crossway point between the cervical spinal cord and the medulla. And there's a very important event that's occurring here, and that is this idea of decussation, crossing the midline. So we have one pathway called corticospinal. It gets to this cervicomedullary junction. And 85% of the axons cross the midline, which now means that your right cerebral hemisphere is going to be influencing your left motor neurons and vice versa. 85% of them cross. And because now the pathway is split, we have to give them sort of more distinct names. So we go from a singular pathway called corticospinal to two pathways, lateral and anterior corticospinal. 100% of the laterals are crossed, and the anterior corticospinals remain ipsilateral, that is, on the same side. And we're going to see how they actually influence different parts of the body in just a second. So this is a critical juncture. Because remember how I said that everybody likes to cross the midline? This is where it's happening for motor activity. The majority of that descending motor activity is crossing the midline, 85% of it. So when you ask the patient to wiggle their left toes, you're really activating their right cortex. And when you ask them to move their left hand, you're really activating the right cortex. Because we're going to see how these extremities are influenced specifically by lateral corticospinal. So it's this crossing event. Not 100% of them cross, rather it's an 85 to 15% split. The majority of them are crossing to form lateral, and a smaller number are staying to become anterior. These terms, lateral and anterior, are merely referring to their relative position of these pathways within the spinal cord. Their relative position. We'll see that. So, here we actually have, just to remind ourselves about the spinal cord and what we're ultimately hoping to do here. So we know that within the ventral part of the gray matter of the spinal cord, we have our motor neurons. Those are the ones we're going to affect. These motor neurons send axons out of the ventral root to go on to innervate skeletal muscle. Now, we can take this spinal cord and section it and get this really nice appearance of the tissue again. Now, I always sort of preface what's going on here by this statement. So, early anatomists found very early on that you want to preserve human tissue for people who look at histologically, anatomically, right? And they happened upon this one fixative. I'll tell you the name, but don't worry about it. It's called osmium. And osmium is very lipophilic. And osmium turns myelin black, right, in color. And so you know that this mantle around the spinal cord is all the white matter. Well, it's just turned the white matter dark. Nothing more than that. So when you look at these images and you say, well, hold on now, the white matter is the darkest thing there. It's just the stain, okay? It's still white matter, which makes, again, this inner core of gray matter relatively dull by comparison, okay? Now, when we look, again, if you think about now, I've said how we can just very easily cut the spinal cord in half, a dorsal half for sensory, a ventral half for motor. We can now even get into the motor aspect and say that there's a more medial and lateral component to it. That when you look at that ventral horn, there's a medial aspect and a lateral aspect, and there are neurons segregated in there that we're going to see do very different things. Just to remind ourselves that when we're thinking about the hand, we're talking about the cervical spinal cord. When you're thinking about the leg and the foot, you're thinking about the lumbosacral spinal cord. And in between, you don't really have a heck of a lot going on, right? You just have the little intercostals, right? So if you think about the spinal cord itself, you get these terms referred to as the cervical and lumbosacral enlargements. And it's just referring to the fact that physically, the cervical and lumbosacral spinal cords are physically bigger than the thoracic spinal cord. And they're physically bigger because you need many more neurons in here to do this than you need neurons in here to just move your little intercostal muscles. Makes a lot of sense. So these enlargements refer to the fact that the size of the spinal cord in the cervical and lumbosacral regions are absolutely larger because you need to cram in there many more neurons to do the more precise things there than you need for the thoracic spinal cord. Okay, so here is that cervicomedullary junction where all 85% of our axons are going to cross the midline. And because these are huge bundles of axons, it actually distorts the appearance of this tissue right in this area. So here's one of these big bundles coming from the right over to the left, and just behind it would be the left coming over to the right. They're crisscrossing like this. Now there's a term here that is absolutely important that you understand. and that term is pyramidal. Early anatomists, for whatever reason, thought that the cortical spinal pathway here, when you see it in cross-sectional view, kind of looks pyramidal, and the term has stuck to the fact that you may go to clinic and someone will say to you that this patient has a pyramidal lesion versus an extrapyramidal lesion. And pyramidal is referring to corticospinal pathway. And we think about this crossing as being the pyramidal decussation. Okay? It's an old term to specifically describe the corticospinal pathway. So that's where pyramidal comes from, and the idea of decussation is crossing. Left to right, right to left. All right. So if you look then at the other cross-sectional view of the spinal cord here, we're not doing anything with the dorsal aspect because that's all sensory. We just want to do the ventral aspect because that's motor. And if you look here, we can now see in that ventral spinal cord two divisions. There are medial motor neurons and lateral motor neurons. Remember, these neurons are now going to be received. These are the lower motor neurons that are going to be receiving the information from the upper motor neurons. Do I fire or do I not fire? And if you look, they actually have different targets. Lateral is all about limb muscles. Out here, lateral. And if you think lateral, like it's to the outside of, that's what is going on here. So lateral corticospinal is about the lateral motor neurons in the ventral horn, and it's about your limb muscles. There are other motor neurons that are affecting the axial muscles, more the midline, right? Your shoulder, your hip, things right at the midline. And these medial motor neurons clearly then are different from the lateral motor neurons, because to do this, right, takes a lot more than just simply shrugging your shoulders or moving your arm around. So, we get into this idea of a somatotopic organization. So, here are our lateral and our medial neurons. And there's the representation of the body. Look at this. So, here is our limb, our hand, our wrist, arm, elbow. And as we get closer to the arm and the shoulder and the axial muscles, right, we're actually going from a lateral to a medial, outer to inner. It makes perfect sense, right? Even physically, where these neurons are located in the spinal cord, we're going from the outside to the more midline. And that's exactly how they're represented on the human body, out to in. And if you think lateral versus medial, well, you only have to worry about lateral. So in the lateral, motor neurons that are located here are receiving direct inputs from who? Lateral corticospinal. And these medial neurons in here are receiving inputs from anterior corticospinal. Now, so does everybody see that? Now, this is a really nice representation of the schematic. So imagine in here, right, in this part of the spinal cord, are thousands of motor neurons sitting there receiving these descending cortical inputs. You're going to fire or not fire, right? You're going to activate that skeletal muscle. If you want to have that incredibly precise activity, then maybe having more of a one-to-one relationship is much more important. So these lateral corticospinal axons are only innervating these lateral motor neurons out here for very precise movement of your fingers, even the wiggling of your toes, right? More precise activity. So it's definitely on the same side. But look about these other pathways. So for the more medial aspect of the body, they're receiving information from the left and the right side of the brain. And it's not that big of a deal because it's really not all that precise. And maybe that sharing of information is a way of lessening the load, because this is much more interesting than the merely shrugging of your shoulders, right? So we end up having these two systems, these lateral motor systems and medial motor systems, The lateral affects the distal limbs, and these medial ones affects the axillary muscles, the ones closer to the midline. So who are these pathways? Well, lateral cortical spinal is definitely affecting those lateral motor neurons. So these schematics are the really great representation of how these neurons are organized from the cortex, where their axons go in the brainstem, and how they ultimately pass from the brainstem into the spinal cord, and what they do. Why is this important? Because voluntary motor activity in human beings is incredibly important, right? It's pretty well everything we do. So you have to know as a clinician, if someone tells you that you have a lesion in the midbrain, or in the brainstem, or up in the cortex, or in the spinal cord, that you have a patient present with some sort of motor deficit. And you have to have a general idea about where these axons are passing. So this is just the first of only a few pathways we're actually going to talk about, because these are the really big, clinically relevant ones. And again, this is not a neuroanatomy course where we'd be doing all the different pathways within the brain. But this one is definitely a very important pathway. So we have to recognize that those upper motor neurons residing in the primary motor cortex send their axons down to the internal capsule. We are going to see, if we do the brainstem, how they're associated to the levels of the midbrain, the pons, and the medulla. And then importantly, if we're talking about affecting precise motor activity of the hand, precise motor activity of the toes and the foot, we're talking about the lateral cortical spinal pathway, the one that crosses all their axons from one side onto the other. And of course, we know we can exactly see the exact opposite image from left to right and right to left. It's these lateral cortical spinal axons that innervate motor neurons situated in the more lateral aspect of the ventral horn. And it's those neurons that then innervate the skeletal muscles for the hand. So this is this is the first, and yet is probably up there as the one or two most important motor pathways, right? And that you absolutely have to know. Now, we do know that there's anterior cortical spinal there as well. But anterior cortical spinal is doing something a little different, right? It's actually following exactly the same pathway, right, as the laterals did. It's just that when it gets to that cervicomedullary junction, the axons don't cross the midline then. But rather, they wait until they get to at the appropriate level of the spinal cord to actually bifurcate. Those axons bifurcate to go both left and right. So the axon here descending on the left side, when it gets to the appropriate level of the spinal cord, bifurcates to innervate neurons both on left and right to influence the midline structures. Now, this schematic shows you this idea of a lateral motor system and a medial motor system. And what I'm going to tell you is the only one you need to worry about in the lateral motor system is lateral cortical spinal, this one here. Its helper pathway is this one here called the rubrospinal pathway, the red nucleus, which originates in the brainstem, the midbrain level of the brainstem, and it also helps out. But if I show you this great chart, which makes things pretty clear, I love this, right? This statement here. So rubrospinal gives us all the interesting stuff. Function uncertain in humans. Therefore, you don't need to worry about it, right? Clearly, it helps out lateral cortical spinal, but don't worry about it. So, we have our descending lateral cortical spinal projections. We know that left influences right, right influences left. And we're talking about voluntary motor activity. And that anterior cortical spinal, left is staying on the same side all the way until it gets to the level of the spinal cord, where the axon then bifurcates and both goes left and right to those most medial motor neurons. Now, Anterior does have a couple of helper pathways, vestibulospinal and tectospinal. And this is a good time to actually tell you about how anatomists come up with these terms. Corticospinal describes a pathway. It's a two-part word, corticospinal. Why do we have it? It's because it tells you where the neuron originates and where its axon terminates. It starts in the cortex, terminates in the spinal cord. And once you know that rule, you can then look at any pathway and understand it. Vestibulospinal, vestibular nuclei, okay, ends in the spinal cord. Rubrospinal, I don't know, rubronucleus, I don't know what it is. Red nucleus, sure. Starts in the red nucleus, ends in the spinal cord. Right? So that's how we've come up with these names. These two helper pathways are really going to help again with the midline function. So what I really want you to do as some work tonight is really look at these charts because it's got everything there. So there are 5 things that are absolutely important to know. It's the name of the tract, the site of origin, site of decussation, level of termination, function. Rubrospinal, don't worry about it, especially when it ends on this note, uncertain in humans. That cannot be very good. Whereas lateral corticospinal, there's its name. Lateral refers to its relative position in the spinal cord. It's laterally situated. Corticospinal referring to the fact that the neuron is found in the cortex and its axon terminates in the spinal cord. Where in the cortex? The primary motor cortex, Brodmann area 4. Where do the axons decussate? Well, if it's lateral corticospinal, all of these axons decussate at the cervico-medullary junction in this thing called the pyramidal decussation. Where do they terminate? Well, they terminate throughout the entire spinal cord. There are motor neurons at every level of the spinal cord that require this regulatory cortical information. To move your neck the way you want, you need information. To move your limb, upper limb, lower limbs, you need that information. Intercostal muscles, you need that information. So, we're descending all the way through. When we think about lateral corticospinal, it's really about the extremities of the limbs. It's really at the cervical and lumbosacral enlargements. And what does it do? It's the movement of the limbs. Which limb? Contralateral. Because they cross the midline. So the left motor cortex influences the right limb. The right motor cortex influences the left limb. How about for anterior corticospinal? And we'll stop there. Anterior corticospinal. There's its name. Anterior, relative position in the spinal cord. Corticospinal, cell body is in the cortex, terminates in the spinal cord. Where are these neurons found? Primary motor cortex. Great. Site of decussation. They don't really decussate, right? They don't decussate. Rather, when they're in the level of the spinal cord, they bifurcate. What are they doing? Well, if you think about the axial muscles, right? It's really cervical and thoracic. And then we have control of what? The bilateral axial and girdle muscles. So upper arm, right? The shoulder, your hip joint, bilaterally. It's influencing both, the left influences both left and right populations of neurons. So I'll finish up. There's a break now. You have another lecturer. I come back. So we'll finish up here, and we'll be able to do a few clinical cases.